what we think, the reason we think you remember what you had for breakfast this morning is because your brain is different from this morning. If you had the same brain you woke up with physically, I mean, you would actually be the same physical person, right? You'd remember nothing from today. Okay? So this is what we think. We think that memory is somehow associated with some physiological change in your brain, right? So just pointing at areas and saying this does that, is really unsatisfying, right? It's like, well, what does it do, right? So long-term potentiation is seems to be a big part of memory, and it seems to be one of those physical changes that is associated with memory. Okay? So we think maybe this is the physiological process that happens when you create a memory. Okay, so what is long-term potentiation? This is persistent, use-dependent increase in the efficiency of a synaptic transition. So this is persistent, and it's use-dependent. So it lasts a little while, right? Persistent, and it's use-dependent. It depends on your experience. So depending on your experience, this thing lasts a little while, and it's a change in how your two neurons actually communicate with each other. Okay? So the efficiency of synaptic transition. Okay. So, and you remember that a synapse, here's your synapse, right? So there's your presynaptic cell, there's your postsynaptic cell. The presynaptic cell fires, neurotransmitter gets released into the synaptic cleft. That neurotransmitter then binds to a receptor. That receptor opens, a channel, ions flow in, and that depolarizes our cell. And if it depolarizes it enough, it makes that cell fire. Right? So this right here is the synapse, right? The junction between the pre and postsynaptic cell. And so we're saying that the efficiency of the synapse will increase. What does that mean? That means this cell will be better able to drive this cell. So it makes this cell more likely to fire. Okay, so let's talk about that. Oh, and this is really popular because number one is it seems to be theoretically important. So it's considered to be a substrate of memory. And also it's very easy to do in vitro. So you can use rats, slices of rat brain and actually elicit LTP. Like the neural substrate, like the physical change. Okay. So what does a typical LTP experiment look like? So it looks like this. And usually they use the hippocampus because the hippocampus is a major memory area, right? So we're going to use the hippocampus. But this could be other areas as well. Okay. And so the hippocampus kind of looks like this. Here for a second. The hippocampus kind of looks like a swirl. Remember that? If you cut the frontal view, it kind of looks like a swirl, right? So there's an area called CA3, and there's an area called CA1. And CA3 has these neurons that send these long axons over to CA1 neurons. And here's our CA1 neurons, and they have these dendrites that kind of hang like this. And so our CA3 neurons send information over to our CA1 neurons. So this is the cell body, this is the axon leaving, and this is the terminal buttons over here, right? So this is the synapse right there. Okay? Good so far? Now, if you get confused, you have to stop me. Okay? We have the whole time for this. Right? So just please stop me if you get confused. Okay, so we call these the Schaefer collaterals. these axons that run through CA1. Okay, so what they do in the typical LTP experiment is they take a baseline. So what they do is they pick one of these neurons, right? Let's pick this one. And we present, we give it a little electrical stimulation, right? And it makes it fire, okay? And so we make the cell fire. This information goes down, neurotransmitters released, right? And what we do is we do an extracellular recording on this cell here. So we record this cell, okay? And so the idea here is we give a little electrical stimulation and we see how efficient is this synapse to begin with, right? So we see how well does this cell actually drive that cell. Can this cell make this cell fire? Make sense? Okay, yeah? All right. So that's baseline. So at baseline, we're simply trying to determine how efficiently does our CA3 cell actually drive our CA1 cell. Okay? Now what we do is we try to induce LTP. How do we do this? So remember LTP is this change in efficiency at the synapse, right? So the way we try to induce LTP is with a tetanus, and we give a huge electrical stimulation now, and we do it like 100 times really fast. We go, right? And that makes the cell like fire like crazy, right? And so a lot of neurotransmitter gets released onto our CA1 neuron, right? And so we just really, really drive the cell. Just the one cell. Yeah. So we're just talking about this cell here. Okay? That's it? So far? Okay. All right. And then what we do at test, after that happens, we give the same electrical stimulation that we did at baseline. So we give the same electrical stimulation, and we try to measure the resulting excitation in CA1, right? So if we increase the efficiency of this synapse by doing the LTP thing, by giving the tetanus, what should we see? What do you think we should see? More efficiency, right? We should definitely see more efficiency. In terms of our CA1 neuron, what that means is you'll see more excitatory postsynaptic potential in response to this same stimulation that you got at baseline. Because this is the same exact stimulation. So in other words, you have this cell, and you have, and this cell fires, right? So this is my terminal button, right? This is the end of my axon. And here's our postsynaptic cell, right? When we first gave stimulation, this fired maybe at a level of 5. I don't know, 1 to 10, it's about 5, right? Random number. But then after LTP, after this tetanus, same stimulation, right? Didn't change anything, didn't change the stimulation. Now all of a sudden, this cell will respond to it with like a 7 or an 8, right? A bigger excitatory postsynaptic potential to the same exact stimulation. Yes?
Is that because of like more receptors or more transmitters?
We'll talk about it. Yeah. Yeah. I just want you to understand the phenomenon first. Yeah? Yeah, you get it. Right? Okay. Is that good with everybody? Yes. Okay. So the result would be that it elicits a bigger excitatory postsynaptic potential from our CA1 neuron. Okay. So this is interesting because this is a change. This is a change that number one is persistent. So it turns out, like, how long does this last? Might be one of your questions, right? How long does this actually last? It lasts could last seconds, could last minutes, could actually last hours, could last years. So this could be a persistent change depending on how you set up your experiment. Yeah?
How does the efficiency affect individual rates?
We'll talk about it. Yeah. So number one, you got this persistent change. And also recall that this change depends, it depends on experience, right? So there's natural ways you can drive your neurons. But really this change happened as a result of something, some incident, right? In the natural world, this would be caused by some incident that happened, right? So it's experience dependent, right? It depends on the experience. We didn't do anything weird biologically, right? We just stimulated. We just made this one fire a bunch, right? And then we got this change, and the change is long-term. It's input specific. So here's one question. Well, we know that it actually increased the efficiency here, right? Because I did baseline and then I did a test, right, after the tetanus, right? So we know that it increased the efficiency here. Question. Maybe it just made this neuron more sensitive altogether, right? Maybe if we stimulated here, this neuron would just respond in general to everything more, right? That doesn't seem to be the case. So it seems to be input specific, meaning if I drive this neuron here, yeah, there's a lot of inputs going to this particular neuron, right? But it seems to be that the tetanus actually makes this synapse more efficient. It doesn't make every synapse more efficient. It's just this one now that has the ability to drive this cell more. Okay, so it's input specific. Is that good? Okay, so this is what that's showing you. This is kind of a more realistic version of what it would look like. So our one CA3 neuron would actually send tons of input. And there would be another one. What they're showing you here is this is baseline, so you put a little stimuli, and this is how the cell responds, right? But after the tetanus, it responds more, because this one, we got the actual tetanus, right? And here's our other input. At baseline, it responds as much, right? But after the tetanus, it still responds as much, because this one didn't actually engage in the execution. So it's input specific. Is that good? It's just a more complicated. Another interesting thing, and this is where we start to get into learning, is turns out you can cooperatively induce LTP. How does this work? So let's say we have our CA3, our CA1s, right? And let's say we know for a fact that if we did, let's say, a medium stimulation 100 times, that's not enough to induce LTP. We really need that big, strong stimulation. Okay? But it turns out, if I gave a medium kind of tetanus here and a medium tetanus here at the same time, so simultaneously, so let's say this happens simultaneously, right? If we do this, then together they can actually induce LTP at both of their synapses. Okay? So, a weaker kind of stimulus can actually induce LTP in both synapses. So what would be like the cognitive example of this? So, a cognitive example of this would be, so I eat, like, the way I like ice cream, I like to get what is that called? The yellow vanilla? What is it called?
French vanilla. I like to get French vanilla, and I like to put tons of cinnamon on it. That's what I like. And then I like to eat it, right? So here you have this idea of vanilla, right? And you have this idea of cinnamon. And each one now, like when I think of cinnamon, can actually make me think. It can elicit the idea of ice cream, right? So when I think of vanilla, I can actually, or when I think of cinnamon, it can make me think of ice cream. So because these two have been paired multiple times with this one idea, because cinnamon and vanilla have been paired and have been driving the idea of ice cream, suddenly now one of these can actually trigger the idea of ice cream. Okay? So two ideas that have been presented over and over again in the stimulus can actually elicit this. So here's the book example. If you don't like my example, you can go with the book example. The book example gives this idea that the idea of a rose. So the memory of a rose, what can trigger that? Well, a rose is always paired with a particular smell, and a rose is always paired with a particular sight, right? So you could actually, because those are simultaneously presented over and over again, eventually one of those, just one of them can elicit the idea of a rose. Okay? So this is two weak ideas that can be paired simultaneously to bring up one idea, right? All right. And that's interesting. And there's a lot of learning phenomenon on that idea that weak stimuli can actually cause some learning. But I actually think the next idea is a little bit more interesting. But that's my personal preference. So here is the idea that it's associative. This is hugely interesting. So here, with associativity, a weak stimulus may be insufficient to cause LTP, right? So you have a super weak stimulus. But if it comes simultaneously with a really strong stimulus, that input will actually receive the benefit of the LTP. So, in other words, this we know would not elicit LTP. My little small stimulation. We know for a fact that won't induce LTP. However, if we present this simultaneously with my big input here, right? If this neuron really fires, right? This one will have the benefit of LTP. So here you get LTP. This synapse becomes more efficient, right? But in addition, because this one was simultaneously presented with this big tetanus, this will also receive the benefit of LTP. And yeah.
What if it was like two?
Oh yeah. It's just for yeah. So whatever input is simultaneous with that big input, they'll receive LTP. So what's going on there? So where do we see this behavior? So let's get out of the hippocampus for a minute and let's get into the amygdala. There are certain things that can drive your amygdala, right? If I show you a picture, if I show you a snake, your amygdala will light up, right? People tend to have this natural kind of response to snakes, right? If I shock you, your amygdala will light up. So when we do our experiments on rats, the rats don't like the shock, and you can see it in their brain. Their amygdala will light up, right? So you have this biologically relevant stimulus that can drive fear in the amygdala. So I'm not in hippocampal world anymore. I'm in the amygdala world, right? And what happens is it's actually really important for you to remember the things that precede biologically relevant events, right? So it's not hard to make a rat learn that if they get a tone shock, it's not hard for them to learn. They'll learn pretty quick to fear tone, right? The rat will freeze when it hears a tone because the tone predicts a shock, right? So this thing that is not biologically relevant at first, that does not have the ability to drive the amygdala by itself, if it's presented with this really strong biologically relevant stimulus, right, then it's actually, if that happens over and over again, then it itself has the ability to drive this fear response. Does that make sense? So this is actually the way they think Pavlovian conditioning happens at the cellular level. Because this is a big one, and this is a small one. The other one, yeah, it's two middle ones. Yeah, it's two medium ones. Yeah. Same thing though, really. Same phenomenon. Yeah. Does that make sense? Yes? We're good?
I'm sorry.
Yes.
They're both simultaneously led. So it's
Yes.
just the difference is medium, small.
Yes. Yeah. So yeah, this is both simultaneous. So simultaneous. Okay. Yes? Good? Alrighty. It's going to remind you a lot more of it. Okay. So, you thought it was done, right? Now this is when we start talking about ions and stuff, right? So what is actually going